It says here, inconceivable. In mammals, pretty much every cell in the body carries two sets of genes. One set comes from mom, the other from dad. Eggs and sperm are the only exceptions. They have just one set. Then when a sperm fertilizes an egg, those two sets combine, restoring the usual number of two sets per cell. Gormley and other same-sex partners are currently barred from their dreams by a phenomenon called genetic imprinting. It uses a distinct tag from each parent to mark the DNA that mammals pass on to their offspring. The process ensures that for a small percentage of genes, we only express a copy of the genetic material provided by our mother or our father. When this imprinting process goes awry, kids end up with an inactive gene regions that cause miscarriages, developmental defects, and cancer. It says during this genomic imprinting, mom's distinct collection of tags typically turns off certain genes so that just dad's copy is expressed, and dad imparts his own marks that leave only the maternal copy on. Most imprints silence gene expression, but some activate it. That's a problem with same-sex couples who want to have a baby. If both sets of offspring's genes come from maternal DNA, for example, then both copies of imprinted genes will be off, so the embryo can't make any of the gene's products. We don't get the full set of gene products that we need to undergo proper development unless we have both maternal and paternal contribution to the fertilized egg, says Marissa Bartolome, a geneticist at the University of Pennsylvania in Philadelphia, who discovered one of the first imprinted genes in mice. And then they said scientists discovered genomic imprinting in mammals about 30 years ago. During experiments in the mid-1980s, researchers removed either the maternal or paternal genetic contributions from newly fertilized mouse eggs. Then they transferred in a second set of genes from another mouse to create embryos with either two sets of female genetic material or two sets of male genetic material. A surrogate mouse was able to gestate the embryos, but none survived. The findings showed normal development requiring genetic material from both a father and a mother. More than that, the outcomes revealed that the maternal and paternal genetic material differ from each other in meaningful ways. Later experiments revealed mice develop differently depending on whether they happen to receive both copies of certain regions of DNA from one parent rather than one copy from each parent. Mice with hairpin-shaped tails were telling examples. When researchers deleted the gene region responsible for a hairpin tail from a mother's genome, mice embryos grew large and died partway through gestation. In contrast, deleting the same region from paternal genome had no effect on the rodent's growth or development. In the 3 decades since, researchers have found more imprinted genes. They suspect there are about 100 to 200 such genes, and the molecular tags that silence them. Scientists have also taken strides connecting imprinting defects to developmental disorders in humans, but all along researchers have known that imprinting prevents same-sex parents from having children. And then it says editing out impossibility. In October of 2018, researchers overcame this impossibility in mice by deleting imprinted regions. Why lie? And a team of the Chinese Academy of Sciences in Beijing produced healthy mice from 2 moms. The researchers also created mouse pups from 2 dads for the first time. However, the offspring died just a few days after birth. Despite the loss, Lai is optimistic. The research shows what is possible, he says. To overcome the imprinting barrier, Lai and his fellow researchers turned to CRISPR, a genetic editing technique that's made altering genomes easier than ever. They used the tool to delete gene regions from embryonic stem cells from mice mothers. The researchers then injected those modified stem cells into the egg of a female mouse and then used a third surrogate female mouse to carry the fetus to term. The team had already seen some success 2 years earlier when they created mouse pups with 2 genetic mothers by deleting the 2 imprinted regions. Although these bimaternal mice also grew to adulthood and produced pups of their own, they developed growth defects. On average, the bimaternal mice were 20% lighter than their heteroparent counterparts. In their latest study, Lai and his team also deleted a third region from the mother's gene, which restored the animal's growth to normal. It says, but the scientists had to clear a few more hurdles to generate mice from two genetic fathers. They found, though, a process of trial and error that they needed to remove twice as many imprinted regions in the bipaternal mice as the bimaternal mice. In total, the team deleted 7 imprinted regions to successfully create mice from 2 dads. Still, the numbers were not in their favor. Only 2.5% of the embryos made it to term, and less than half of 1% lived for 2 days. None made it to adulthood. The produced bipaternal mice are not viable, which implies more obstacles are needed to cross to support their postnatal survival, if possible, Lai says. The lower birth rate, on the other hand, implies the existence of an unknown barrier hindering the development of bipaternal embryos. In contrast, bimaternal mice fared much better. These mice grew to adulthood and were healthy enough to have pups of their own by mating with typical male mice. They also behaved the same as the control mice. As far as the researchers could tell, the bimaternal mice appeared as healthy and as normal as any other laboratory mice. It says it does not mean that they are normal in every aspect, Lai cautions. One cannot investigate all the aspects under restricted experimental conditions, which a limited number of animals. So basically what it sounds like to me is that they made and had a huge success in the mice, that they were able to create pups, mouse pups, from taking the genes from 2 moms and then inserting it into a surrogate mother. And then that mouse pup was able to grow up to adulthood and then have their own children with male mice. Whereas the mice that was produced with the genetic material of 2 dads died during the process of being created in the surrogate mother. So tell me, what do you think about this? Do you think in the future that it will be possible for same-sex partners to be able to have their own children that share both of their genetics? Or do you think this is something that is not going to happen at all whatsoever? Anyways, please like, share, and subscribe. I'm out.